Okay, so should I just start? I think Marian is running late, so he might just get on with the seminar. I will not attempt to introduce everybody. We'll just be presenting the research that was presented in form of posters at a conference in Zakopane about high-performance computing implementation of different frameworks. And I'll just go first. Right, so my research is about, my application of that I'll be presenting is high throughput statistical shape modeling of venous anatomy using containerized defermetrica on ARIS supercomputer. So the context is deep vein thrombosis in the lower limb. It is influenced by anatomy and flow disturbances, but the link between shape and hemodynamic risk metrics remain poorly quantified. So statistical shape modeling provides a way to characterize population level anatomical variability and combined with flow simulations to quantify the link between shape and flow. SSM is quite computationally demanding, so to support reproducible large-scale experiments, the workflow was migrated to ARIS supercomputer and executed in a containerized environment. And this is the full workflow overview. In this presentation, I'm only focusing on the implementation aspects, but this is context. So shapes of here, the common iliac veins are extracted from medical images, and these are full 3D shapes, and they're 2D projections in the plane of a typical angiogram. And then they're processed through CFD simulations to extract flow metrics of interest in studying thrombosis, and in parallel, they are fed to the statistical shape modeling framework using defermetrica. And there we're using 2 steps, deterministic atlas, which computes a template shape for a given set. And then the template is used when running so-called principal geodesic analysis, which decomposes our set of shapes into shape mode components. We also run CFD on these, and at the end, we correlate computed flow metrics and shape modes. There's a preprint of a paper available. We're currently going through the review process. If you're interested in the actual results and the correlation and the CFD outputs. But here's the technical problem. Defermetrica is the most commonly cited SSM tool. It has extensive documentation, but it is relatively old. And when I first approached it, it turned out to be an iceberg of problems. Biggest are fragile dependencies. This is a Python library. So I used Conda on WSL Ubuntu. The environment setup, the documentation provides no longer works. The dependencies could not be resolved. Specifically, Torch for CPU kernel usage had to be installed manually because the required version is no longer hosted on Conda repositories. When everything finally installed, I actually had to tweak the source code because of outdated string handling. And this meant commenting out a couple of lines here and there. So I made the installation work finally on my laptop, only to find out that my PC does not have enough memory for 3D SSM simulations because SSM performance depends on a well-converged atlas template, computing the atlas template that's of nice shape. This requires extensive parameter exploration and significant computing resources, and this has 2 aspects attached to it. So the number of shapes considered in the base set is relevant, and the number of control points in space that encapsulates the set is also relevant. The shape deformations are computed with respect to these control points, so the more you have, the more computationally demanding it is, but you need to have enough of them for the simulation to converge. So I managed to run 2D simulations eventually on a workstation, but in 3D it was impossible, even for a small set of 12 3D shapes. So to address the installation issue, I packed Defermetrica into a Docker container. This created an environment with all the necessary dependencies and tweaks in the source code that could be transferred between machines without too much hassle. And to address the computational issue, we shipped the container to Ares supercomputer, converting it from Docker to Obtainer. And the files setting up the simulation parameters and execution scripts were fed from the outside of the container. The simulations were parallelized via Slurm job arrays, and typically the best outcomes came from using 15 CPUs with 20 to 50 gigabytes of RAM. The efficiency and runtime was quite difficult to predict because it depended on the model parameter set used, and their behavior was quite difficult to predict. But as I said before, the number of control points was, of course, a strong predictor of the needed resources. So in the end, we ended up with an improved throughput of the SSM pipeline, where we could systematically search the parameter space. And at the end, I can add that I used the CPU version, but there's also a GPU option in the Informatica, which I eventually managed to install as well inside the container. But I did not manage to make it actually work more efficiently than the CPU version, which must be something with the setup of the Informatica, but I really needed to get a paper out, so I did not have time to focus on that. So this is the technical aspects of that research, and this work was part of the Preludium project financed by National Science Center. And of course, we used the Ares supercomputer on CipherNet resources. Thank you.
Okay, I am the next one, and I will share my screen right now. It should be visible. Yes, I hope everybody hears me well. So the title of the poster is Benchmarking a Heterogeneous Metagenomics Pipeline on Ares and Helios Supercomputers. This work was done in collaboration with Kinga Zielińska, Michał Orzechowski, Karol Zając, Tomasz Kościółek, and Maciej Malawski. So a few words of introduction. We have developed a metagenomics pipeline which processes publicly available SRA samples available from NCBI NIH. We convert it to desired format, then we do pre-processing, remove human reads, perform assembly with tool called MegaHit, and then we have three branches. The pipeline characterizes by very heterogeneous requirements. Many steps have high resource requirements. On the other hand, other steps have very need a lot of memory in hundreds of gigabytes, which makes difficult to optimize well, and this is quite challenging but doable. We developed it originally for Ares supercomputer, and our goal was to migrate to a newer and better Helios supercomputer, which was made publicly available a few months ago. We wanted to analyze the performance improvement between the supercomputers on a meaningful batch, and also find possible optimizations for large-scale processing. That was our goal in the research, scaling from 100 samples to 100,000 of those RNA sequences samples. So the main difference between the Helios and Ares supercomputer is the difference between the processors used within the clusters. We can see that we have more cores on the Helios supercomputers, and also, which is very important, the available memory per node, because higher memory allows us to better allocate our resources for the memory-intensive steps, which makes Helios very suitable in our scenario. The main challenges with the pipeline is that the tool called BACTA frequently accesses 80 gigabyte database file, and we utilize the MMFS solution to significantly improve the processing speed, even 10 times using this approach. But on the other hand, we need to allocate enough memory for the job. And also we need to have enough storage space for intermediate files within the that are processed and created during the pipeline execution. So we have conducted an experiment on both supercomputers with the processing of 100 samples, which is about 150 gigabytes of fast queue data. As you can see, Helios is about 50% faster than ours. Similarly, the CPU usage is CPU hours usage of Helios is better than the RS, and it shows that this is a better supercomputer, newer, and we get our results faster, even though the efficiency is lower. But this is mainly due to I/O nature of the pipeline steps. Thank you.
Okay, so I will be next. Can you see my screen?
Yes.
Okay, so my name is Carlos Zając. I am a scientific programmer at Sano, and today I would like to present our work on automating CFD workflows on HPC systems using PyFluent. And I would like to acknowledge that this work was a collaboration. Magda was responsible for developing the CFD models, preparing simulation setups, and performing the scientific analysis of the results, while I was contributing more in a technical way. So a quick background. CFD simulations are widely used in engineering and scientific research. And at Sano, my colleagues are running some CFD simulations of blood flow. And as a computational resources continue to grow, more and more studies require not just a single simulations, but like entire simulation campaigns that involve parameter studies. And for this, we need a computational power of HPC infrastructures. However, a major challenge here is that many CFD workflows still remain highly interactive. So the typical Fluent workflow often involves manual geometry preparation, some solver configurations through graphical user interfaces, and also interactive post-processing. And while this approach works well for some individual simulations, it becomes very inefficient and difficult to reproduce when we are talking about hundreds or thousands of simulations required to obtain some meaningful outcomes. So this slows experimentation and becomes complicated at scale. And it is an issue, especially for the HPC, because here we usually do not have user interface. We have to transfer the input-output data from pre and post processing between our local machines and the cluster. So this end-to-end automation of such a CFD workflow is a challenge here. We noticed that that there is a research gap and only few solutions are integrating CFD pipelines with UQ tools. And this motivated us to investigate how Fluent simulations can be automated on HPC. So the main goal was to minimize this manual intervention and enable scalable execution. From the key ideas, we wanted to just take a simple CFD workflow and try to script it, then programmatically enable the simulation control and setup for the runs. Also a batch execution of multiple cases on HPC cluster, so the matter of submission, and also to prepare for the larger experiments like parameter studies. To achieve this, we built a semi-automated workflow based on technologies shown here. So the approach builds on modern HPC and Python ecosystem tools. We have already tested them on Cyphronet's ARES cluster. And the most important here is PyFluent, which provides a Python interface to ANSYS Fluent. Then we have Slurm RM jobs. Instead of launching simulations manually, simulations are executed in batch mode on HPC resources through this mechanism. And while batch execution is implemented using those RIs, the workflow can also be integrated with Dask distributed and EasyViewQ for more complex sensitivity analysis and uncertainty quantification studies. And those are tools that we are using to support VVUQ experiments and workflows, also in Gemini project. So yeah, we have developed demonstrative use cases showcasing UQ capabilities and incorporating those optimization techniques. I will not delve into all of those specific tools, but yeah, it illustrates the high potential to run experiments at scale in most scenarios. And those are very easy and accessible mechanisms. And now let me briefly walk through the workflow. So it all starts with the definition of simulation parameters that we want to explore. So we can either generate them with EasyViewQ or just use a simple CSV with cases that we want to be processed. These cases will be run for a specific geometry models, which require the preparation, of course. And it is one of the most difficult parts to automate. And it cannot be done fully for all input formats because yet PyFluent is not supporting that. So there other libraries can also be applied to provide the geometry that ANSYS Fluent is capable of meshing. And based on this input data, we are submitting an array job. Then it goes through the meshing and solver setups that is being done by PyFluent, and mesh generation and simulation are executed with ANSYS module on the ARIS cluster. And once the simulations are completed, the results are collected and post-processed automatically. Here, PyFluent also allows to extract some data and metrics from the runs. But because the workflow is script-based, it can easily be extended with some custom pre-processing and post-processing tasks. Yes, and as I mentioned before, the array job approach here can be substituted with task if more dynamic resource allocation is needed. So one of the key advantages of this approach is the scalability, because by leveraging HPC resources, we were able to perform simulation campaigns that will be impractical on local machines. In our studies, the workflow enabled the execution of nearly 1,000 simulations involving systematic perturbations of both boundary conditions and patient-specific geometries. Automation not only reduced the overall computation time through this parallel execution, but also significantly reduced the effort that was required to prepare and manage the experiments. And of course, this allows researchers to focus more on analyzing the results and less on the repetitive manual tasks. As for the future plans, because PyFluent provides these powerful automation capabilities, but is not yet fully stable in terms of API consistency and feature coverage, we need to wait for this maturity here. In certain stages of the workflow, such as geometry preparation for complex models or also extraction of advanced simulation metrics, we still require some further development here. So these areas represent the future work. Here are references, and with this, thank you for your attention. If there are no questions, the next probably is Taras.
Yes.
Yeah.
So I would like to present our PubDB application that was developed by scientific programmers team, in particular by me, Taras Zhehulin, and Adam Nowak, Jan Meisner, and Piotr Nowakowski. So let's begin with our motivation to do it. Sano is thankfully growing, and the amount of our publications is rising every year. That's great, but we need to keep track of them in order to settle research grants, to calculate our efficiency, our performance indicators, and also to present our successes to others, to increase visibility of our institution. So to make this correctly, to make this solution scalable, we need a shared, consistent format for the publication records, and the reporting of the publications should be simple and convenient to encourage researchers to keep track of their publications and not forget about this. Before, it was facilitated by Excel document where we all had to enter our publications, but it was shared both for
moderators, for management, and for regular researchers. So there were a lot of columns, and it wasn't really clear which columns have to be, which columns are obligatory, which are not obligatory, which are for the moderators, and we should not touch them as simple researchers. So it was quite a confusing experience to use it, but now it's our PubDB application, which provides a convenient user interface for it. So its main features include role-based access via Microsoft Entra ID. So it is a secure access system which automatically delineates access, showing for the regular researchers from Sano only the fields which are intended for them, and for moderators the additional fields for only for moderators. And it is designed for multiple users, also for external users who can browse the publications, authors, see the statistics without actually changing something. So it is secured from the users from the outside, and it allows for advanced search and filtering to conveniently browse through publications and see what actually interests you, to narrow your list of publications to the ones that that you want to see. And there are also a set of administrator tools for moderators, which simplify the process of reporting and calculating KPIs. Additionally, there is a statistics dashboard, which is generated by our publication tool. It is presented on Sano's main page, and it shows some nice graphs about our efficiency. You can see the amount of publications by year, by research group, and some other interesting statistics. This is facilitated thanks to API that the application exposes for our web page, where the statistics are shown. There are also some future features that we are developing at the moment, and you will see them soon, such as Dataverse integration. If your publication has a dataset attached to it, and this dataset is published on our Sano Dataverse in Roadbook Federation, then the PubDB application will automatically detect this and show a nice widget allowing to cite the dataset easily, to download some files from it directly from the application interface. Additionally, the advanced dashboards and reporting are incoming for the moderators, so that they can easily generate reports and evaluate the efficiency of teams or particular authors. And the integration with external publication databases is planned, for example, with Google Scholar, Scopus, or Web of Science, so that even if you forget to add your publication to the database, it will be retrieved automatically, verified by the moderators, and then added to the list. So if you would like to see more features and try it yourself, please visit the application. If you have any suggestions, please write on Slack channel, PubDB Slack channel, and feel free to contact me for additional questions or suggestions. Thank you very much.
So I will start. My name is Dominika Ciupek, and today I will present you my research evaluating the stability of deep neural networks for brain microstructural parameter estimation under varying diffusion gradient schemes. Bipolar is a chronic psychiatric condition characterized by significant fluctuations in mood and energy, with diagnosis often delayed by 5 to 10 years. The diffusion-weighted magnetic resonance imaging offer promise for identifying objective biomarkers in psychiatric assessment. In particular, diffusion tensor imaging enables the analysis of brain microstructure, which may help distinguish bipolar disorder patients from healthy controls. However, standard acquisition protocols and evaluation methods remain insufficient for ensuring clinically meaningful accuracy. In most cases, microstructural parameters are treated as natural images and evaluated using standard metrics such as mean squared error or mean structural similarity index measure. And those metrics focus on image similarity rather than clinical relevance. And therefore, this study aims to evaluate the potential of deep learning models to accurately estimate microstructural parameters while preserving clinically relevant differences between bipolar disorder patients and healthy controls. In our study, we used two microstructural parameters, radial diffusivity and fractional anisotropy. Fractional anisotropy was essential for performing the tract-based spatial statistics analysis. We used the UNet, a deep learning model architecture that is commonly applied in medical imaging. We trained the model in 3 different configurations based on the number of gradient directions, 64 directions which were corresponding to the original acquisition, and 6 and 3 directions which were simulated from the original data using spherical harmonics. In each configuration, we trained the model 100 times with different weight initializations to evaluate the stability of the training process. Instead of relying solely on the standard metrics, we also performed the tract-based spatial statistics analysis. And this kind of analysis allowed us to detect the statistically significant differences between bipolar disorder patients and healthy controls. Training deep learning models at this scale is extremely computationally demanding, and therefore the choice of the infrastructure is a critical factor. Training a single model on the local computer takes around 121 hours, while running the same task at the RS CPUs reduces this time to only 21 hours. However, the real improvement comes from using the GPU acceleration, and training the model on the Athena GPU takes only 35 minutes, which means that it was more than 200 times faster than on the local supercomputer. And those differences became even more striking when we consider the full analysis, so running 300 models, because on the local supercomputer, it would take more than 4 years of continuous computations. And on the supercomputer Athena, when we run around 40 scripts at the same time, it takes only 4.5 hours. And these results demonstrate that exhaustive analysis of the AI stability are not feasible for the local machines, and that the access to high-performance computing resources is essential for them. If we look only at the evaluation metrics, all of the models performed really well. The mean structural similarity index measure is really high. It's equal to 0.95 for only 3 gradient directions. And also the mean squared error values are relatively low compared to the overall range of the data, which was from 0 to 1. However, the TBSS analyses show the true hidden cost of reducing the number of gradient directions. The false positive rate for the free number of gradient directions increases dramatically to over 350%. And that basically means that the model is detecting statistically significant differences in areas where there are none of them. And only for the 64 gradient directions, the false positive rate level is reasonable. And these findings highlight important limitations of natural image metrics in medical imaging. They provide a misleading sense of accuracy while hiding the true errors. And the visual analysis confirm our observations from the numerical results. All of the models correctly detect the true positive voxels, but for the 3 and 6 gradient directions, we can observe bright and stable clusters of false positive voxels, which means that the model is not randomly generating error, but they are consistently seeing statistically significant differences in incorrect regions across multiple runs. And based on our analysis, we can draw some conclusions. deep learning models achieved high performance based on standard image similarity metrics. However, these metrics did not reflect clinical reliability. Variations in model weight initialization had little effect on evaluation metrics, but resulted in significant differences in the TBSS analysis, and it was presented in the form of standard deviation of each of the metric. And this underscores the necessity for stability analysis in medical imaging studies. And large-scale stability analysis was made feasible through GPA-based parallelization, reducing computation time from years to hours. And that's it. Thank you very much for your attention. And if there are no questions, probably Elena could present next.
Hi, so I'm Elena, and I'm doing an internship now in Sano in the extreme scale data analysis team. And I'm going to present the using high performance computer for the application of pins in cardiovascular models, a poster that I presented in KDM 2026 conference. Okay. So first, the purpose and objectives of my project. First, we are going to use pins, which I'm going to explain later, to personalize cardiovascular models so that they fit an individual patient without manual parameter tuning. We also were aiming towards adding complexity to the models while keeping hemodynamics true. That means not violating any physical laws while adding this complexity. And then the ultimate purpose was to generalize across multiple models and patients so that this pin architecture can be applied to more scenarios. Okay, so first, cardiovascular model. This is a model of the cardiovascular system that consists of heart and blood vessels, veins and arteries, and it has been going around for years. It has been used to measure specific parameters of the cardiovascular system in a zero-dimensional way. So, for example, pressure translates to voltage in electrical systems, valves translate to diodes, and capacitors translate to heart chambers. Heart chambers can be either atrium and ventricles, both right and left. And as you can see on the right, we see the electrical circuit analog to a four-chamber cardiovascular system, which four-chamber consists of two atriums and two ventricles, left and right. And as you can see, there are many parameters involved in these kind of models. The more chambers you have and the more accurate you want to be, the more parameters to be personalized for each patient. So there is a simpler version depending on what you want to measure. And here we have, for example, a two-chamber version. On the right, you can see the electrical circuit scheme, and we only have the left part of the heart and only the systemic flow. We neglect the pulmonary flow. And that leaves us with only, yet like a lot, 10 equations and multiple parameters to be fit, but way less than a more complicated four-chamber model or so on. So these would be the 10 equations over time. As you can see there, it's periodic because each period corresponds to one heart period, one heartbeat. And yeah, you can see pressures, volumes, and flows over time. And we depend on initial conditions and parameters that feed these equations. So each set of parameters will give different variables over time, and that would correspond to a specific patient. For example, if a patient has a bigger heart, you would, for example, increase or decrease the compliance of the capacitors. Now, the purpose of the pins here would be to add complexity. As you can see, the red lines of this left ventricular volume over time would correspond to a one-chamber model, so the simplest one. The blue ones correspond to a two-chamber model, and the green ones to a four-chamber. The more chambers and the more complexity we add, the more steep are the derivatives, and more delicate is the function, so we grasp more hemodynamics that might be important for clinical purposes. So the purpose of this pin is to add complexity, hence increase the derivatives or decrease them, and match magnitudes. So that would be part of the personalization process by modifying the derivatives of the ODE system, the ordinary differential equation system that I showed before of 10 equations. Now, what exactly are physically informed neural networks and what is different from classical neural networks? The key is in the loss function. The loss function includes a physical constraint that we know must be met, and that makes them more controllable and understandable, as well as easier to train, as we don't need so much data because we know that they must satisfy some conditions, and that makes it converge towards the solution faster. And the NN will change the ODE system by adding, that's very important, its additions to the derivatives that already exist. And it will calculate these additions so that the derivatives approximate better the more complex, for example, four chambers analog. So it would look kind of like this. As you can see, they are additions. They are in red in the equations. So we have the derivatives, and the neural network would calculate the additions corresponding to these specific derivatives and parameter set. And it would kind of look like in an electrical circuit as having an extra component that is kind of like a black box, but not so much black box as a classical neural network, because we kind of know that it's following physical constraints that we know must be true. This would be the architecture of the neural network, the physically informed neural network. We insert a set of parameters and the equations. The equations would always be the same for these kind of applications, but if they would be other applications, the equations could be different. And then the neural network calculates the additions to the derivatives. Then it solves these equations, having a time series of each variable, and then it compares them to a target that would be the experimental data of each patient, and depending on how much the loss is, then it backpropagates and calculates the NN additions again. And by the end of the training, we should have an optimal ODE plus addition system that keeps the simplicity of and little parameters of a two-chamber model, but has steeper derivatives and more hemodynamic complexity. These are some of the losses that I tried. So you can see the data loss, that's the most typical, that's no physically informed, but it would be the predicted against the target. Then we can also compare first derivatives, because I said that they are important to capture hemodynamic complexity. Then we have the physics loss, that is directly implementing the ODE again, and see if the new predicted ODE system plus additions satisfy still the equations of flow in, flow out must equal difference in volume and those kinds of equations. Mass conservation, so no flow, hence blood must appear or disappear during a period. Zero mean would make this not drift over time, because if the additions to the derivatives accumulate, this may cause the signal not to be periodic anymore, and that cannot be. We also know that volumes must not be negative, never. And then we enforce periodicity, because we are taking into account a healthy, stable patient, and we don't want heartbeats to be different from one another over time. So this poster also presented some results. We tested some different loss combinations against single variables. On the left, you can see the variables that they were implemented into. And then the last row would be the NN contributions implemented to all variables, and on the columns you can see the different loss combinations. So we have a different types of results, positive or negative. The ones on red means the loss is increasing, so those are not nice, but we can see some green ones, and that was very positive. And here would be one of the best trainings of all. We can see early stopping after 100 iterations, because the loss was increasing a little bit too much. So, but we can see very positive results on the right. Pin must equal data, and that is blue equaling green. And as you can see, in the first four variables, counting from top left, are pretty matching the green one, so that's very positive, and they are further apart from the red. However, on the last two variables, it's not so much the case, but anyway, this was considered a successful training. However, it was seen that when tested against other patients, meaning other sets of initial conditions and parameters, the results were not so nice, because the neural network learned the same additions for every patient, not so much adjusting to each patient. So that's leaving us with future work. The plan is to try with multiple patients and one target function to see if it could be more personalizable and leading to every patient to the same experimental data, meaning that all models would match the one experimental data of a real patient, and that would eventually lead to personalization. But that's still ongoing. This was not presented on the paper. So yeah, this is basically what I just mentioned. And the second next step after the first one would be to generalize this, capturing all the data and training a neural network, not PIN, so that it learns how each model would be generalized to one experimental data, and then we could make this generalizable to more patients. Suggestions are very welcome, as this is still very fresh work. Thank you.
Yeah, so Marian sends his apologies. He was originally going to chair this meeting, but his car broke down, so he's stuck at a shop. So sorry for that. The next seminar will, I believe, take place in 2 weeks' time. Yes, the topic will be announced on our website. It's still a matter of discussion who will be presenting, but yeah. If there are no further questions, then I think we can close the seminar today. So I will thank you all for participating, also on behalf of Mariam.
Thank you very much. Goodbye.